So today, we're diving into a really fascinating new hypothesis that could completely change the way we think about our own DNA. It's called the Genomic Coherence Threshold, and it's a concept from researcher and author Kevin L. Brown. Let's break down what this idea is all about and, more importantly, why it could be a real game changer.
We're going to walk through this in 5 simple parts. First, we'll look at the problem, the big mystery our current genetic models are struggling to solve. Then we'll get right into the tipping point hypothesis itself. After that, we'll talk about how you could actually measure it, how you could try to prove it wrong, and finally, we'll wrap up with why this idea matters so much.
All right, so first things first, let's talk about the big puzzle we're facing. We have these incredible tools for looking at our DNA, right? But there's still a huge piece of the picture that's just missing. I want you to think about a bridge for a second. You can add more and more weight to it, car after car, and for a while, everything seems totally fine. But you know there's a limit. Eventually, you hit a tipping point, and the entire structure is at risk. So the question is, does our genome, probably the most complex information system we know of, work the same way? And this really gets to the heart of the problem. On one hand, we are fantastic at studying the small stuff, individual genes, single mutations. That's the micro view. But where we stumble is explaining the big picture. How does the entire genome, all 3 billion letters of it, hang together as one stable system? How does it handle all that stress over generations? And that's where this new idea comes in, trying to answer that exact question. It's the tipping point hypothesis. Okay, that's the core idea, straight from the research. So what does that actually mean? Well, put simply, it's saying that our genome is constantly collecting stress. Think of it like a pressure tank. This pressure comes from mutations, from our environment, all sorts of things. And if that pressure builds up and crosses a critical line, the whole system is forced to change. It has to adapt, reorganize itself, or, well, it starts to lose its stability. Now, the key word in all of this is coherence. What is genomic coherence? The easiest way to think about it is this. It's the state where the genome is working like a finely tuned machine. All the parts are integrated, everything is running smoothly. It's that stable, healthy state we want our DNA to be in. So the hypothesis breaks this down into a 3-phase cycle. It all starts in the base phase. Everything is in balance, stable. Then you enter the pressure phase. This is where strain starts to build up from all those different sources. And finally, you get the integration phase. This is where the system has to do something to resolve all that pressure, either for better or for worse. It's a constant, dynamic process. Okay, this all sounds like a really neat idea in theory, but how can we actually see it happening? I mean, how do you measure something like genomic pressure? Well, the pressure comes from a few key places. You've got new mutations coming into the gene pool, or maybe our DNA repair systems just aren't as efficient as they used to be. You could also have a mismatch between our ancient genes and our super modern world. And that last one, weaker natural selection, basically just means that some of the natural forces that used to filter out small errors might not be as strong today, so those errors can pile up. Each one adds a little more strain to the system. So, how would you go about measuring this? The process is actually pretty logical. First, you'd measure all those inputs we just talked about that create pressure. Second, you'd look for the warning signs of instability across a whole population. And third, you'd combine all that data to calculate the total pressure and see just how close that system is to its tipping point. And what do these warning signs look like? They're not abstract ideas. We're talking about real, measurable trends. Things like an increase in certain developmental disorders, or maybe you start seeing dysfunctions all clustering together in very specific biological pathways. That clustering is the key. It suggests it's not just a few random problems, but a system-wide issue. Now, we're at what I think is the most important part of any new scientific idea, figuring out all the ways you could prove it's totally wrong. And you have to ask that question, right? A good idea has to be more than just an interesting narrative. It needs to be testable. It has to be, well, breakable. And that's the crucial point. For a hypothesis to be genuinely scientific, there has to be a way to falsify it. You have to be able to stand up and say, if we observe this, then my whole theory is false. And this is exactly what's laid out here, the specific conditions that would completely invalidate this hypothesis. For instance, if you saw a population under huge genomic pressure for generations and nothing happened, the hypothesis is wrong. If our current genetic models could already explain everything perfectly, we wouldn't need this new one. These clear, testable criteria are what make it a real scientific model. So let's bring this all home.
We've got the problem, the hypothesis, and how to test it. Now for the big question. So what? Why should any of us care? What are the real-world implications here? Well, for one, this could lead to much smarter genomic medicine. Imagine, instead of just looking at one gene at a time, doctors could actually assess the overall stability of a person's entire genomic system. It could also give us a brand new way to understand aging. Maybe aging is, in part, just a slow loss of this genomic coherence over a lifetime. This model could give us the tools to actually find out. And this is pretty wild. The idea isn't even limited to biology. It could potentially apply to other really complex information systems, like helping us predict when a huge AI model might become unstable or start to drift away from its original purpose. Ultimately, the big shift here is moving from looking at genes like individual car parts to viewing the entire genome as a whole system that needs active, ongoing maintenance to stay coherent and functional. And that leaves us with a pretty profound question to chew on. With all the amazing tools we have today for gene sequencing and data analysis, are we on the verge of becoming the first generation that can actually measure the overall coherence of our own species' genome? That is a pretty big thought.